Yeah, thanks for the invitation to join this discussion today, and it's really nice to see so many people interested in this area, and the field has really progressed over the years. And thanks obviously to Christian and Rachel for the wonderful presentations that really lay the groundwork for some of the stuff I'm going to talk about today. So hopefully that'll make it digestible to this audience. So I was asked to give a sort of research update on MAGEL2 biology. This is going to be very basic biology, and I'm specifically going to talk about insights into a recent study that we completed into how also MAGEL2 alters the brain. And this study was recently published. This is the sort of the title of that study, and it was a real collaboration between a number of groups spanning many academic institutions, as it highlighted on the right there, and also was benefited from funding from the FPWR Foundation. So thanks to those folks and for everyone contributing. So my lab has been working on the function of MAGEL2 probably for about 10 years now. And some number of years now, back in 2013, we published the first report of a function of MAGEL2 in regulating how protein abundance in the cell is controlled by the presence of MAGEL2. And this diagram is here to sort of point out sort of the high level thinking of what we know about MAGEL2 in this process. And specifically, if you think about in sort of normal cells in normal individuals, what we know is that proteins, which make up most of our cells, are controlled in their abundance and localization, such that they need to be in certain parts of the cell to function properly. And for example, here is just a protein that in blue, you can see all these little squiggly lines are a specific protein. It needs to be localized in this certain pattern as you see here. And what we found is that if you delete MAGEL2, which is of course what's happening in the context of Prader-Willi syndrome or Schaaf-Yang syndrome on the right, you see instead of the pattern on the left and sort of normal individual cells, now you have this pattern where instead of the blue protein being out here on this black line, this is indicating the plasma membrane, it all accumulated intracellular and end up going into the trash can, which we call the lysosome in the cell. And so basically the fundamental discovery from this work was that MAGEL2 regulates the cellular protein trafficking and the abundance of a specific subset of proteins within the cell. And that is interesting because that maybe is sort of provides the basis for the underlying symptoms that you see presented in your children is due to this alterations in the abundance and localization of proteins like these shown here in blue, okay? And we followed up that study with a more detailed understanding of how this works, and I won't go into it because it's not really relevant to the discussion today. But we showed that this MAGEL2 protein works with other proteins, some of you've already heard about, in what we call a complex here called the MUS complex, composed of these three proteins. And it regulates this pathway through a process that Rachel described to you called ubiquination. And I really won't say anything more about that for the remainder of my talk, but just know that that's something that we know MAGEL2 is doing, okay? Now today, what I'm gonna focus on is really trying to understand what proteins, what are the blue things here that are really regulated by MAGEL2. So the objective of the study I'm going to describe is um, is determine if aberrant protein trafficking upon loss of MAGEL2 contributes to or is causal to the clinical symptoms presented in SYS and PWS. Okay, and we wanted to really take unbiased approaches to determine this, and I'll show you what we did in this way. And what we did is specifically in this study is we wanted to identify those proteins whose trafficking in abundance, those little blue squiggles I showed you before, which of those depend on MAGEL2? And specifically, we're particularly interested in the hypothalamus, as already highlighted by Rachel and Christian, as a important a part of the brain where MAGEL2 is functioning. The second is we wanted to then extend those studies that are primarily driven by work in the mouse to understand them better in other neuronal models, particularly human neuronal models, to see how those extend into a more potentially relevant setting. And then the final is, of course, if we can determine the pathological relevance of what we find in contributing to these diseases. And of course, this is, of course, the hardest and still ongoing work. And I'll just say up front, sort of the conclusion of the work I'm going to show you is that loss of MAGEL2 in Schaaf-Yang syndrome or PWS leads to defects in the production of small proteins or molecules that we call neuropeptides in both the MAGEL2 knockout mouse in the hypothalamus, as well as in human neurons that lack MAGEL2. And we believe this likely is contributing to the clinical phenotypes. And I'll describe to you what these neuropeptides are and whatnot coming up in the next several minutes, okay? So how did we start this study? Well, we started the study again, wanting to take a really unbiased approach, systematic, where we said, let's just take the mice that are either normal, wild type, or those that have lost the MAGEL2 gene, specifically that paternal copy that Christian elegantly described the genetics of. And this is sort of a model of Schaaf-Yang syndrome, and you heard a lot about that from Rachel, who pioneered the development of this mouse model. And we said, let's take these two mice, and let's take tissues from these mice. The hypothalamus, of course, as you've heard, is of primary interest. Hypothalamus regulates other endocrine organs like the pituitary. Let's take other metabolic organs, liver, adipose, other parts of the brain, etc. And let's just do unbiased analysis of how the proteins change between the normal state and the Schaaf-Yang state in the mouse. And when we did that, the hypothalamus was particularly enlightening. And this is a graph that sort of shows the relative abundance of thousands of proteins. Each dot here, each of these gray little circles is an individual protein that was detected by an approach called mass spectrometry in the tissue from these mice. And what you're looking at is the relative abundance of these proteins between the wild type mouse and the SYS mouse. And, you know, when we got this data, it was really one of those prototypical, you know, eureka moments that you probably think of as for scientists is happening all the time, where, you know, you do some work and then boom, you figured it out, right? But in reality, that's not how science works. It's a slow slog through the mud, and only on rare occasions do these type of just results that, you know, knock you over the head as obvious come along. And this is one of those. And why is that? That is because all these sort of gray dots they don't really change that much. There's only a very small number of either green, those proteins that are decreased in the SYS mouse, or an even smaller number of proteins shown in red that increase in the SYS mouse. All the other thousands of proteins really don't change significantly. And what's even more important is that all these over here that I've highlighted that are going down in the knockout mouse, those are all part of the same pathway, and all part of the same process. And this, again, rarely happens when you do these type of unbiased studies where the result is just so obvious, that it doesn't take a real genius to figure out what's happening. And it turned out that all these proteins were part of this neuropeptide um, processing and production pathway, and I'll describe that in the next few slides. So this pathway is really downregulated in the hypothalamus of these Schaaf-Yang model mice. Okay. So this slide shows you sort of what are neuropeptides. So as I mentioned, these are small protein-like molecules or peptides that are used by neurons to communicate with one another. Okay, and they're used throughout the nervous system. They're used in other endocrine ways throughout the body, and they regulate many brain functions. Some of them are very related to what you've been hearing about: food intake, learning and memory, social behavior, metabolism, reproduction, pain sensation, analgesia, etc. And some of them you may know by name. For example, oxytocin. Oxytocin is a neuropeptide. And so here I've just listed some others, okay? And these neuropeptides basically are secreted by neurons. Here is a neuron in gray. They're secreted by neurons in the hypothalamus as well as other cells in the body in a very regulated way. They're actually packaged into little vesicles in the cell, and that's what you see here in green circles. We call these secretory granules. And these vesicles concentrate these neuropeptide hormones such that they're ready to be released when the cell is ready for them to to release them. And when they get released outside the cell, they go and act on other cells in the body. That could be neighboring neurons or other places. Okay. And I sort of have a little more information here about these secretory granules, these little vesicles. You saw those green dots. And I'll say here that these are vesicles inside the cell that process and concentrate those neuropeptides into these very small packets that can be regulated so that once the cell receives some kind of external stimulus, they can burst out these neuropeptides and therefore signal to other cells in the body. And these secretory granules are composed of a very small number of proteins, and primarily the neuropeptides I've referred to, like oxytocin, but also the proteins highlighted down here. And there's sort of two flavors. There are these over here that are called granins. These are blue ones, and they basically concentrate the neuropeptides. In this diagram, the little red things are the neuropeptides, and the granins are clustering those neuropeptides all together. And then there are these proteins that are sort of on the outside here, and these are called proteases. They clip the neuropeptides into their mature form. It turns out the peptides are made in an immature form that are not active. Only once they get into these proper location in the secretory granule do the proteases, like these proteins called PCSK1 or PCSK2, do they then clip these things into their mature active form. And I highlight these because we're going to be following through the rest of the talk proteins like the granins, like chromogranin B, or the prohormone convertases that process these neuropeptides, PCSK1 and PCSK2, okay? And so here is just some data that followed up that initial observation to really confirm that we were right. So you're looking at the levels of these three proteins I just highlighted, PCSK1, PCSK2, and chromogranin B, all parts of the secretory granules. And you're looking in mice, either the wild type mice in gray or the SYS MAGEL2 knockout model in red. And you can see at different age of the mice, here's 2, 4, 8, or 20 weeks. In all cases, the abundance of these secretory granule components, PCSK1, 2, and chromogranin B, are all downregulated. And so that's consistent with our initial observation that I told you was so obvious, and so this reproduced this in a large cohort of mice. Now, that's in the mouse hypothalamus. What about in humans? And you've been introduced to this earlier in Christian's talk of what we call induced pluripotent stem cells, or iPS cells, that can be taken, that you can take cells from a human and reprogram those cells into stem cells, and then those stem cells can be differentiated in all different types of cells, including neurons from the brain. And so we said, well, let's try to validate whether what we find in the mouse actually happens in a human. So we took iPS cells that are wild type, and we genetically engineered those cells to delete MAGEL2 as well as the rest of those genes in the Prader-Willi locus, that chromosome 15 region that Christian described to you. And we said, with that model, do we see the same thing that we saw in the mouse hypothalamic tissue? And indeed, we did. This is just, again, looking at those same proteins I described before, PCSK1, 2, or chromogranin B, as well as another one, CPE. And in gray are the wild type iPS cells that have been differentiated into neurons. And in red are iPS cells that have been differentiated into neurons but lack MAGEL2 and the other Prader-Willi genes. And you can see here that, again, all these proteins are down regulated in these cells that have been deleted of MAGEL2, confirming that this is indeed happening in human neurons, not just in mouse. So that's great. iPS cells are great, but the one issue with iPS cells is you have to start with, say, skin cells and go back to a stem cell state and then differentiate into neural cells. And all this requires various manipulations that may not be accurate to what actually happens in the body. And so there's one closer way to get to what's happening directly, and that is using these dental pulp stem cells. It turns out within the deep in the root of your teeth, there are cell types in here called dental pulp stem cells, abbreviated DPSC, that are indeed true stem cells that can be differentiated into neurons or other cell types. And upon extraction of these, or just the teeth normally falling out, these are typically the baby teeth, you can crack open those baby teeth and extract those cells, put them into a culture, and differentiate them into different cells without any type of genetic manipulations, okay? And so we were able to do this with our collaborator, Larry Ryder at University of Tennessee, and show again that the same thing that we were observing in the mouse, as well as the human iPS cells, shown here again for these three proteins for consistency, PCSK1, 2, and chromogranin B, are all decreased in neurons derived from these dental pulp stem cells in the case of Prader-Willi patients or SYS patients. Importantly, this is a control of a unrelated syndrome, Angelman syndrome, in green, that are not affected. So this really validated that indeed this secretory granule neuropeptide pathway was disrupted both in mouse hypothalamic neurons lacking specifically MAGEL2, but also in two models of neuronal models in human iPS cells or dental pulp stem cells that are differentiated into neurons. So I told you that those neuropeptides are packaged in these secretory granules that include those PCSK1, 2, and chromogranin B proteins. So if those proteins are decreased, we would expect to see lack of those secretory granules, those little green dots, if you remember back to that original cartoon. And so it turns out you can image these things in a experimental setting. And so here is tissue from a wild type mouse or a MAGEL2 knockout mouse in the hypothalamus. And I've pointed out here with these little yellow arrows, you can see these dark black structures. These are actual secretory granules. And what we find is that when we count up these secretory granules in a wild type hypothalamus compared to in the knockout, MAGEL2 knockout, you can see a substantial reduction in the number of these secretory granules, consistent with what I've been showing you. And this was also true in the other models I described. So in the human iPS cell model, where we knock out MAGEL2, we can see a reduction in these secretory granules. Same is also true in those dental pulp stem cells, where depletion of MAGEL2 results in decreased numbers of these secretory granules. Here you can see these little green dots are secretory granules. This is in the control individuals, but neurons derived from patients lacking MAGEL2, you can see that there's much less of these little green dots. So that's great. Secretory granules are sort of the vesicles that are delivering the material, and the material that's key within them are the neuropeptides. And so we then wanted to determine whether the neuropeptides were decreased also. And indeed, that's what you see. This is looking at 5 different neuropeptides: oxytocin, AVP, SST, GnRH, and MSH. Looking through a proteomics approach, mass spectrometry that I described at the beginning, of wild type mice in gray and the knockout MAGEL2 knockout SYS model in red. And you can see in all these cases, there's decreased abundance of those neuropeptides in those mice from the hypothalamic tissue. Now that's also true again in our neuronal models. So here is in the iPS, human iPS cells that we've differentiated into neurons. You can again see in this case, oxytocin levels are decreased in those cells. These are just 2 different conditions, not relevant for today. And furthermore, when we obtained plasma from Prader-Willi patients and we measured the abundance of the secretory granule marker in the plasma, chromogranin B, you can see that those Prader-Willi patients had decreased levels of chromogranin B in the plasma compared to their control siblings or parents. Again, consistent that what we're observing in the mouse hypothalamic tissue, as well as the cell culture human neuronal models, iPS cell or dental pulp stem cells, are consistent with actually what's happening in humans. So this brings me to the sort of conclusion of what I wanted to tell you about today from this study, is that we believe that MAGEL2 in the healthy individual is important for proper trafficking of proteins, including the production of these secretory granules that carry with them these neuropeptides like oxytocin, et cetera, and particularly in the hypothalamus for producing these and allowing them to have their proper action upon release from those cells. However, loss of MAGEL2 in the context of Schaaf-Yang syndrome or Prader-Willi syndrome really results in the disruption of production of these secretory granules and thus neuropeptides, and as a result, you have less of these hormones being produced. And I would say that there's a lot of evidence supporting our findings. And these are just 3 studies I won't go into due to time that are consistent with what we've been seeing both in other mouse models as well as in human patient cells. And then finally, as Rachel's already mentioned, if indeed you have less of these neuropeptides being produced, one might think that you could rescue some of the defects observed in individuals with loss of MAGEL2 by sort of hormonal therapy, sort of delivering these neuropeptides exogenously. And she's already commented on these studies, so I won't go into detail, but these are 4 different studies, 2 that have been published, 2 that are just being coming out soon, that highlight that delivery of oxytocin, one of these neuropeptides, can have benefit. And I think that's, again, consistent with what I was mentioning, that we believe that these findings are likely contributing to the pathology of the disease, and that correction of those neuropeptides could be a potential way of helping as a therapy. So I'll stop there. I mentioned this is a very collaborative study from my lab, as well as labs from around the U.S., including at Scripps, UTHS, Mayo Clinic, Broad Institute, and Columbia. And I want to thank all of you for your support over the years. Some of this studies could not have been done without you, giving material, samples, etc. And I'll stop there and be happy to answer any questions you have.
I absolutely love to see the collaboration that you just showed. And I appreciate that you showed us that slide, because that was a lot of people and institutions that brought this work together and made it what it was. We do have a number of questions that are coming in regarding these results. I will do my best to present them to you in as accurate a fashion as possible. Okay, so this was a single pathway that has been disrupted and has caused a number of proteins. It decreases a number of proteins, is what we heard. Yeah. So has this pathway been studied previously? Do we know how we might be able to attack that pathway? Mm-hmm. Are there specific treatments that we know of that may be able mm-hmm. to be a treatment here?
Yeah, so it's an interesting question. I think the question boils down to this study implicates this particular pathway for producing neuropeptides through this secretory granule pathway within the neurons as being disrupted. And are there known mechanisms to say correct that or enhance that could be therapeutic? I think that's the question that's being posed here. And I think that not obvious. The most well established would be the sort of, as I mentioned at the end, the sort of synthetic delivery of the neuropeptides themselves, sort of shortcutting the need for the cell to produce them those themselves. So this is, as I mentioned, sort of what was done in the mice with the oxytocin, where they were delivering oxytocin into those mice exogenously and seeing corrections that would, of course, shortcut the need for the cells to produce it itself. As far as whether we could correct the defect in the production of those neuropeptides, I think boils down to understanding better why we're not making them. I showed you a lot of sort of descriptive evidence that indeed they're not being made as efficiently, and we need to understand now why they're not being made properly. And if we understand that into sufficient molecular details, one can start to think about therapeutics that might circumvent the problem.
Mm-hmm. You played a very active and important role in this discovery. Will you continue to work with collaborators to move this forward into potential treatments, or is this a point where you now hand it off to others?
Yeah.
Where does expertise lie?
Yeah, so for a long time now, I've been doing academic science. Initially at UT Southwestern in Dallas, Texas, and then more recently at St. Jude Children's Research Hospital in Memphis. And that's where most of the work that, or all of the work I've been describing to you has been done at those institutions. Recently, just a few months ago, I've sort of taken a jump in sort of the career track to industry, and specifically now working at Amgen, which is a sort of large pharmaceutical company. And that move is driven by a number of reasons, and I won't get into all those. But you know, I felt like I have been asking the questions of why do things happen, how do things happen, and it was time to sort of ask, more directly as to how our understandings can be translated into to helping people. Much of what we do in basic research is aimed at the discovery process, the sort of intellectual knowledge, with the hope that in 10 or 20 years it might translate into something that is beneficial for society. And I felt like I had done that, and the accomplishments were substantial, and I was ready to now have a more short-term impact. And so I've taken a position at Amgen to lead a new department on development of therapeutics. Now, it's not focused on this disease area, and I don't imagine that my lab will completely, vacate working on MAGEL2 and Schaaf-Yang Prader-Willi, but it'll likely be reduced compared to what it was, just given the difference in direction.
Right. So what can we as the Schaaf-Yang community do to help advance research and ensure that treatments are brought to market as soon as possible?
Well, I think your main objective is to get the most number of people interested as possible. When I say people, I mean researchers. The more labs that are working in the area is going to directly translate to new ideas, new perspectives, more basic science and preclinical translational studies, and all that will of course feed to clinical trials. And you really have to have that before you can move ahead. And I think we're doing a much better job than we were 7 years ago, and even before. I mean, Rachel's been working on this much longer than that, and the field is growing, I feel like. But I think with working with FPWR is a great opportunity to have mechanisms to get researchers involved into this area, and I think that's a prime objective to keep the momentum going.
Right. Well, thank you so much, Dr. Potts, for taking time out to join us. I'd like to thank all of our presenters for taking time out of their busy schedules to come and share what you guys have been working on and what you have learned with us. At this time, we are going to move to Zoom meetings. This is so that we can have our happy hour. I've put up the happy hour Zoom ID and passcode for you one more time. As soon as I end this meeting, which I have to in order to start our next meeting, that slide's going to go away. So if you have not written down the information, please do so now. I also emailed it to you. If it's not in your inbox, check your junk mail. But I hope that you'll all join us over at our meeting so that we can see each other's faces and get to know each other just a little bit better. It's been great having you all here. I'm going to go see you at the Zoom meet. Have a great day.